So it's commonly accepted that Alzheimer's disease is a brain network disease, right? Meaning that the AD pathology advances through specific neuronal systems in the human brain, and it's also well known that that there is a genetic the genetic mutations and genetic variants influence the AD risk. And these two factors come together, obviously, with several challenges for researchers and clinicians. And two of them, in our opinion, is, we wonder if it's possible to detect or visualize the network degeneration in individuals in vivo, right? This is the challenge, especially in our case, using PET imaging. And the other question that we have with this study is, if it's possible to kind of identify biological factors or biological backgrounds or genetic backgrounds associated to those networks that are degenerating in the brain. No? That we don't know yet. So, in order to build new approaches to accomplish those types of aims, we use our 2-year follow-up sample from the Harvard Aging brain study and the PET imaging associated to this study using tau and amyloid tracers, and we developed a novel imaging method that has been specifically designed to extract the network information from the PET neuroimaging. This is based in graph theory principles, and because in the past or previous methods, conventional approaches, in general, they use the intensity of the imaging, of the PET imaging, right, to visualize the deposits, and that's fantastic, but they do not provide the network information that we think underlies, is the basis of the disease, right? That, as I said before, that this, pathological accumulation of tau and amyloid follows specific circuits. So the interesting thing is that the PET imaging can give us that information if we do the appropriate transformation of the data. And this is true, particularly if we use the longitudinal follow-up of the subjects, right, from our population of interest. So if you have the longitudinal information, if it's feasible to obtain, not just the brain, the putative brain pathways of accumulation, but also the directionality of that spreading of pathology. So we tested our novel method in our sample, and we found, for instance, that tau in the temporal lobe produces accumulation of more tau, 2 years later in other, in temporal areas, in surrounding temporal areas, as you can see here, and also in distant areas in the frontal and parietal lobe. We also tested this method using our amyloid images, and we found that, for instance, the posterior cingulate, accumulate, amyloid 2 years later in the surrounding areas and also in the parietal lobe. And in the second part of the study, we investigated the relationship between these types of spreading patterns of pathology of tau and amyloid with the gene expression profiles coming from an atlas of the human brain transcriptome. And we found that our spreading PET patterns were associated with the MAP and CLU gene expression levels in the brain, MAP associated with tau and CLU associated with the amyloid spreading patterns. And we think this is really interesting, right? Because this is kind of one of the first times that we can compare the phenotype of our individuals with some sort of genetic information that might be the basis for the pathological propagation in the human brain. So in summary, our results, we think, open new opportunities to understand the spreading roots of AD pathology in the human brain, and we think that this is an important step, in our opinion, right, to try to transform the way we do the disease staging, because we want to be closer, right, to the more realistic models of AD based on the network nature of this disease.